Okay, I'm actually here with my advisee Anytime, and I want you to say hello Anytime.
Hi.
There she is. She was a little bit nervous about now being recorded in my screencast. She's standing here though, showing me her the dress that she's going to wear tomorrow. Okay, so sorry this is so late, you guys, but we're by Anytime. I want to make sure that you understand this whole concept of membrane transport. So let's cruise over here for a little bit. And I'm going to give you a little bit of a concept map for membrane transport before we go too far. And you should understand that fundamentally there are two different pathways that one could take. There is a passive, meaning no extra energy is needed for it to take place. And then there is an active side where it certainly requires the input of energy for the membrane transport to happen. In the passive transport, we actually have two different types of passive transport. We have something called simple diffusion, and then we also, let me box this up, and we also have the one that I'm going to be talking about a little bit more in just a minute. It's protein-mediated passive transport. Okay? I'll come back to that in just a moment, but let me cruise over here to our active transport pieces. And we have two types of active transport. And the first sounds very similar to the one I just talked about. It's protein, with a dash, mediated, active transport. And then finally, we have what's called bulk transport. And bulk transport actually has a couple of different things. We have, I'll go red here, we have exocytosis, which is also known as secretion. And then we also have endocytosis. However, there are different types of endocytosis that exist. We have pinocytosis and phagocytosis. All right? So let me go ahead and square that up right there for a moment. And then let's return over to the passive transport. Passive transport is driven by molecular motion, meaning that it's the inherent motion that exists in all molecules, whether you're a solid, a liquid, or a gas. And with passive transport, you're also always moving from an area of high concentration to an area of lower concentration. In other words, you're going down the gradient. Okay? And that's why it doesn't require any additional energy for these things to transport materials from one side of the cell to the other. And don't forget, it's not always about bringing things into the cell. Sometimes it's about exporting things from the cell. And then we have active transport. And active transport requires the input of energy, and that energy is in the form of adenosine triphosphate. And adenosine triphosphate is an energy molecule, and we often refer to it as being the energy currency of a cell. You'll understand more about how that works later, but for right now, just know that it's kind of like a rechargeable battery that you can use to do a whole bunch of things. And in the first case, with protein-mediated passive transport, you're moving things from an area of low concentration to an area of high concentration, and that's why you need the ATP, because it's going against the concentration gradient. In the case of bulk transport, the reason why you need ATP is not necessarily because you're moving something against the concentration gradient, but it's because you're requiring the use of vesicles. And the formation of vesicles requires a ton of ATP because there are molecular motors that need to move it around, and then those things need to shape the vesicle, pinch the vesicle, and essentially get the thing to work the way it's supposed to work. And that uses a ton of ATP in the process. Okay, so there we go. That's our overview of membrane transport. Let's, like I said, I want to focus in on this concept of protein-mediated passive transport. So why is this needed? Well, perhaps it's important to understand first that simple diffusion is what we talked about when we talked about osmosis, the movement of water through a selectively permeable membrane. But we can also move things like oxygen and CO2 through the cell membrane without too much trouble. And the reason that this works is that if you remember this phospholipid membrane, let me pause it so I can draw this out. Hold on. Okay, so there's a little portion of the phospholipid membrane. Don't forget that it's a fluid mosaic model, so things are constantly moving around. And in fact, there are cholesterols jammed in here as well, which make it even more fluid. And as a result of moving around all the time, you can't help it. There's going to be these gaps that suddenly form. They close up pretty suddenly, but if they close up in one place, they open up in another. So that means there are constant pores that exist in the phospholipid membrane. And these pores allow for the movement of things, but those things have to be non-charged and relatively small. So if we take something like a carbon dioxide molecule, right, here we have two oxygens and a central carbon. That dude can scooch through in either direction, and it's going to move through from an area of higher concentration to lower concentration through one of these pores. Same thing could be happening with oxygen. So if you have a couple of oxygen molecules, or an oxygen molecule, it can go through in and out according to simple diffusion from an area of high concentration to low concentration. Now that's all fine and dandy, but sometimes you need to move things that are charged and big through another structure. And that structure, in particular in this case, is a protein. And in fact, it's a protein channel or a channel protein that has a relatively big gap in it. Usually they're gated, meaning that they have little caps here that open and close. But essentially what this does is it allows for the charged particles as well as larger particles to be able to scooch through here. So let's say you had a molecule that was this big and it was fairly bulky. It could scooch through, right? And it could go in either direction as long as it's going from high to low. So with protein-mediated passive transport, we're talking about large molecules that can flow through and also charged particles. Now, I'm not going to tell you why charged particles can't go through simple diffusion. That's something that you're going to have to wait for because you guys are working on a group quiz with it. But I think you can understand why large molecules need to go through one of these tunnels, and it's simply because these tunnels can be necessarily larger in diameter than the relatively small pores that open up randomly in the phospholipid membrane. So that way you can let in things that are relatively big or let out things that are relatively big. But don't forget, again, since it's always moving from an area of high to low concentration, we're talking about something that's happening in a passive way without the use of any energy. All right, so there you have it. And again, I want to reiterate one last time that a cell is something that cannot survive without being in its interacting with its environment. And there might be some good stuff out here, positive things. There might be some crappy stuff inside that it needs to get out, and there might even be some crappy things on the outside that it doesn't want to have let in. And so being able to control what goes in and out, letting the good stuff in, kicking out the bad stuff, and blocking the bad stuff so that it can't go in is essential for a cell to be able to survive and continue. You going to bed, cutie? All right, here, say good night. I'm doing a screencast right now. I love you. Bye. Give me a smooch. Bye. Okay. See you in the morning. Bye.
Okay. Oh, should I shut this?
No, that's fine. It's only because they're too loud for Mommy. Okay, crew. That's it. Sorry this is so late. Apologize. Bye.